Welcome to this video on epigenetics in animal populations. We will explore how environmental factors shape gene expression and influence inheritance patterns across different species. In this video, we will define epigenetics, identify its molecular mechanisms, and analyze how environmental triggers drive adaptation, phenotypic plasticity, and transgenerational inheritance. Epigenetics refers to heritable changes in gene expression that do not alter the underlying DNA sequence. Coined by Conrad Waddington, it bridges the gap between the genetic blueprint and observable phenotypes. Unlike genetics, which involves fixed DNA sequences and slow evolutionary responses, epigenetics is highly dynamic. It allows for rapid, reversible responses to environmental stimuli and transgenerational inheritance. Chromatin, the complex of DNA and histone proteins, dictates gene accessibility. Chromatin is loosely packed and active, while heterochromatin is densely packed and typically silenced. The primary epigenetic mechanisms include DNA methylation, histone modification, chromatin remodeling, and the regulatory action of non-coding RNA molecules like mRNA, cRNA, and lncRNA. DNA methylation involves adding a methyl group to cytosine residues within CpG dinucleotides. Catalyzed by DNMTs, this process is generally associated with transcriptional repression. Histone modifications, such as acetylation, methylation, phosphorylation, and ubiquitination, alter chromatin structure and accessibility, thereby directly regulating the binding of transcription factors to the DNA. Non-coding RNAs function as specialized regulators. For example, miRNAs induce mRNA degradation, while piRNAs protect the germline genome from the activity of transposable elements. The epigenome is sensitive to various environmental factors, including abiotic triggers like temperature, nutritional availability, social or physical stressors, environmental toxins, and interactions with pathogens. Nutrition significantly influences epigenetic states. In honeybees, larval diet determines caste, while in mice, maternal nutrition can alter gene methylation, affecting offspring phenotype and health. In many ectotherms, such as turtles and alligators, environmental temperature during development determines sex. This temperature-dependent sex determination is mediated by temperature-sensitive epigenetic modifications. Stress and maternal care also impact development. Studies in rodents show that maternal grooming behaviors can modify the methylation status of key genes, influencing long-term behavioral responses to stress. Pollutants, including heavy metals, endocrine disruptors like BPA, and microplastics, can induce aberrant methylation or chromatin changes, potentially threatening the reproductive success and long-term viability of wildlife populations. Finally, epigenetic variation promotes rapid adaptation, allowing populations to survive environmental shifts. This plasticity is crucial for species living in extreme or changing habitats, such as the Arctic. Let's examine transgenerational epigenetic inheritance. This occurs when epigenetic marks bypass reprogramming during germ cell development, allowing acquired traits to persist across multiple generations, from the F1 to the F3. In social insects, epigenetics drives extreme phenotypic plasticity. Honeybees, ants, and termites utilize differential DNA methylation to create distinct castes from genetically identical individuals, influencing behavior and reproductive morphology. In wildlife conservation, epigenomic profiling serves as a vital tool. It helps assess climate resilience, guides captive breeding to prevent maladaptation, and improves recovery strategies by integrating epigenetic and genomic data. Epigenetics also reshapes evolutionary theory. The expanded evolutionary synthesis incorporates non-genetic variation and soft inheritance, challenging strict neo-Darwinian views that rely solely on DNA mutations and slow gradualism. Case studies illustrate these principles clearly. From the Dutch hunger winter in humans to the agouti gene in mice, where diet modulates coat color, we see how nutrition triggers significant phenotypic shifts. Research relies on powerful laboratory techniques. Bisulfite sequencing remains the gold standard for DNA methylation, while ChIP-seq and ATA-seq are essential for analyzing histone modifications and chromatin accessibility. These techniques have diverse applications. Beyond livestock improvement and aquaculture, they are revolutionizing veterinary diagnostics and helping conservationists monitor stress in endangered species. The future of epigenomics holds both promise and challenge. We must develop AI tools to interpret complex data, refine epigenome editing with technologies like CRISPR-Cas9, and navigate the ethics of modification. In summary, epigenetics is a powerful regulatory system. It bridges the environment and the genome, enabling organisms to adapt rapidly through DNA methylation, histone modifications, and non-coding RNA mechanisms. Thank you for watching this overview of epigenetics. Please subscribe, like, and comment with your questions to continue the discussion on biology and career development.